Hello and welcome to Quarantine Lab. These are my lab assistants, Chloe and Caitlin. Today we're going to be learning about the urinary system in chapter 24. Please follow along in your binder. All right, we will begin by going over the parts in the model. Caitlin is going to assist us through this. All right, Caitlin, where is the kidney?
Right here.
And what does the kidney do?
It filters the blood to make urine.
Excellent. Now, how about the ureter?
Those are here and here.
Yes, and what do?
They carry the urine to the bladder.
Okay, now let's move over to the male and female models. And where are the bladder and urethra in each of these?
The bladder is going to be this portion right here.
And is that the male or female?
This is the female model.
Yes, because here's the uterus right here above the bladder.
And then the bladder for the male is going to be right here.
Yep.
And this is the male urethra. And this is the female urethra.
Yes, very good. So you almost mixed up the vagina with the urethra, which is a common mistake. A lot of people do that. So we have the bladder, which is going to store the urine until you're ready to release it, and then the urethra, which is how the urine exits from the body. Now you can answer questions 2 through 8 on your own using your book, and we will move on to question 9 here, where Chloe is going to demonstrate measuring specific gravity for us. So first I will show you how a hydrometer works. So this is what we use to measure specific gravity, which is telling us how concentrated the solutes are in the urine. So pure water has a specific gravity of 1.000, which is the top line here on this. You put your water in here, you put this in here, and you have to have enough water so that this floats. And you look at where it is floating, what line on here it's floating at, and then you take your reading from those numbers on those lines. The more waste products that are in the urine, the denser that makes the urine. And if the urine is more dense, it makes us float up higher in here, and then you get a higher specific gravity reading. So when you're doing your analysis, if the specific gravity is low, the urine's very dilute, which means the person is drinking lots of water and there's not a lot of waste products concentrated in their urine. If the specific gravity is high, that means they're dehydrated or they have a lot of a particular solute in their urine. So we have here set up pure water, which is 0% salt, some water that's 2% salt, and some water that's 5% salt. And Chloe is going to measure the specific gravity of each of these now. So keep going until it floats. There you go.
It's at 1.00.
All right.
So now I pull these.
Yeah, go ahead and do the next one.
The 2% is at 1.20 and the 5% is at 1.40.
All right, thank you. So as you can see, the correlation here is that the more solutes you have, the denser the liquid is and the higher the hydrometer floats. So you can see as we go along, each hydrometer is floating up higher in the liquid. Now we're going to look at the relationship between hydration level and color of urine and specific gravity of the urine. So we have 2 urine samples here. Both people had 4 milliliters of waste that their kidneys needed to get rid of. One person was well hydrated and was able to put 100 milliliters of water with that 4 milliliters of waste. The other person was dehydrated and could only spare 25 milliliters of water to go with the 4 milliliters of waste. So Chloe's going to compare the color and specific gravity for us.
Okay, so this one is obviously a lot darker. Okay, the light one is at in between 1.00 and 1.1.
Okay.
And the darker one is at about 1.5.
All right. So there you can see how hydration affects the urine. Thank you. Now it's time to do the urinalysis of the 5 bottles of simulated urine. So flip to this page in your binder. We will fill out this chart. So we will begin by looking at the colors. So I have them in order. Here's low, it's clear. There's normal, is pale yellow. High is, kind of dark yellow, orangish. Unknown A is pretty pale, kind of clear. Unknown B is kind of a brown color, brownish, dark color. Now, Caitlin and Chloe are going to test the pH for us. So this is low, has a pH of about 3. Now we'll do the normal. Has a pH about 5 or 6. Now for the high. Has a pH of about 8 or 9. Unknown A has a pH of about 3. And unknown B has a pH about 9. Now for the glucose and protein test strip, you're looking to see if it changes color. So we're going to do one in just plain water. This is just plain distilled water. That's going to be our comparison one. So we're going to use that. And if the strips stay the same color, you guys go ahead. If the strips stay the same color, then that's a negative. So these ones that get dipped in the urine, if the color matches our one from plain water, we know that it's negative, that the protein and glucose was not there. If they change, then that's a positive. That means that either protein or glucose was present. Now these strips have to sit for 30 seconds, so while we're waiting, Chloe and Caitlin are going to get started on specific gravity. Caitlin, which sample is that?
This is sample high, and it's sitting at about 2.5.
All right. And Chloe, which sample are you doing?
This is sample low, and it's about 1.00.
Okay. While they do the next specific gravity, we'll start comparing these. So here is the low. Looks pretty much the same. Here's the normal. Looks pretty much the same. Here is high. Looks like we've got a little bit of color change. Here is unknown A. Looks like there's some color change in the green one there. And here is unknown B. Might be a little bit darker on the yellow one. Okay, we're ready for some more specific gravity. Chloe, which sample do you have?
This is the normal sample, and it is about 1.2.
All right.
This is unknown A, and it is about 1.1, 1.2.
Okay. And Caitlin is doing unknown B now.
Okay, this is unknown B, and it's sitting at almost 3.
All right, thank you. So now that you have all of the data, you can go through and answer the rest of the questions in your binder using the data that we've gathered and the information in the reading section of your binder.